Thank Dr. Shrija Lakshmi for giving this guest lecture on Introduction to Astrobiology. She is from the Indian Institute of Space Sciences and Technology, and she works in the Department of Chemistry. Some of the interesting things that she works on, which she may not have enough time as such today to talk about, but some of the interesting things that she works on are bioastronautics, space life sciences, space medicine, and so on and so forth. But ma'am will talk about introduction to astrobiology as much as she can. For the students, please at the end of the session, you can either raise your hand and ask the question directly, or you can put it in the chat box and ask questions to Dr. Shrija Lakshmi. I'm sure she'll have a lot to share and a lot of insight to provide as well. So with that, Dr. Shrija Lakshmi, the floor is all yours.
Yeah. Thank you, Dr. Pavitra. And good morning to one and all. Here, I wish that the students are all also visible for at least in the beginning of the session. Is it possible for me to see them at least for a while?
Sure, ma'am. So request the students to at least switch on your video.
Yeah. And you just tell your name because only 9 of them are there, right?
Yes.
Before starting, I should know.
Okay. Is anyone students, can you please switch on your camera even for a short period? Okay. All right. Then we have.
So they are in the campus or at home or lab? Where are they?
So, ma'am, most of them are hostelers. So they live here on campus, and when they have lecture, I mean, they are supposed to be here in the building. But most of them are hostelers. A couple of them come from far away.
Oh, okay.
So we have Kushi. We have.
I think they can start introducing. Kushi, can you start introducing yourself?
Yes, ma'am. Good morning, ma'am. I am Kushi Khare, and I am.
Your background is in?
I have done my bachelor's in biochemistry.
Biochem. Okay, fine. Good. Yeah. Sai, Mohan Sai.
Good morning, ma'am. I'm Mohan Sai. Yes, I'm from Telangana, Hyderabad, and I did my bachelor's in biotechnology.
Biotech.
From Osmania University.
Okay.
And yeah. Good to have you here, ma'am. Thank you.
Thank you. So next one. Aditi, I can see Aditi. I'm
we have
calling
Pallavi
the names
also, but it says 1+. So Pallavi.
Yeah, that 1+. I could.
Yes, Pallavi, please.
Yeah. Hello, ma'am. Good morning. My name is Pallavi. I am from Assam, and I have done my bachelor's in aircraft maintenance engineering.
Sorry, Pallavi, what was the branch?
It's aircraft maintenance engineering.
Aircraft. Oh my goodness. Yeah, great. Yeah. Next person.
Aditi.
Aditi, please.
Good morning, ma'am. I'm Aditi, and I did my bachelor's in biotechnology from Kolhapur University.
Okay, great. Okay. Yeah, nice meeting you. Yeah, Nilesh is there.
Yeah, hello, ma'am. Myself Nilesh Bandekar, and I've done my BSc in zoology.
Zoology.
Yeah,
great. Yeah. So how many are there now? 1, 2, 3, 4, 5.
We have Mayuri. We have Renish.
Yeah, because no, I'm writing down your name and your specialization, because we will be moving ahead with some of the activities also. This is not going to be a single time lecture, okay? So we are now joining a course. So I'm expecting you to be along with us, okay, along with your MIT professors. You need to have a lot of networking. That is very important.
Yes.
Okay, that is essentially why I agreed to Sneha when she was inviting.
So we have Renish, Mayuri, Lazi, and Madhu left. Please switch on your cameras and at least introduce yourself. Yes, Renish is.
Hello, ma'am. Yes, ma'am. My name is. Good morning, ma'am. My name is Renish Michael Raj, and I did my bachelor's in. Yes, ma'am.
Renish?
Yes, ma'am.
Okay.
Yes, ma'am. Renish. I did my BSc in microbiology from Mumbai University. My college name was NR Swami, SIWS.
Very good. Okay. Okay. Yeah.
Yes.
Anybody else?
Yes, Lazi, Mayuri, or Madhu, anyone. Yes, Lazi is there.
Good morning, ma'am.
Yeah.
My name is Lasidoma Bhutia. I'm from Sikkim, and I did my bachelor's in medical biotechnology.
Medical biotech. Very nice. Very
Yes,
nice.
ma'am.
Great. Your name one more time, please. I couldn't.
It's Lasidoma Bhutia.
Can you put it in the chat box, please? Because I'm recording it, or maybe later Dr. Pavitra can share the
Sure.
students list with
Sure,
me.
definitely.
Yes,
definitely.
Yeah, yeah. And somebody was.
Yes, unfortunately, Mayuri is one of the
Mayuri is
students who is
traveling.
traveling from, yes, she's the one who travels from out of campus.
Okay, no, no.
So her BSc is in life sciences
Life
from
science,
Mumbai
yeah.
University.
Could see that, yeah. Great.
Madhu.
Who else?
Madhu.
Good morning, ma'am. My name is Madhu. Good morning, ma'am. My name is Madhu, and I am from Visakhapatnam, Andhra Pradesh. I did my bachelor's in botany and zoology.
Botany and so on. Okay, great, 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 great. Okay, so yeah, I think all the nine have sort of introduced.
Yes.
So I'm really happy that we have a very diverse sort of crowd here, smaller one, but still with a lot of diverse disciplines. So this is one beautiful aspect of astrobiology as well. Okay, where we need people of different expertise to come together and to speak a common language. Okay, so to start with, I will share my presentation first. Okay, so this is going to be a sort of a very basic lecture. I plan it in 2 parts. Let us see how long I can continue for the day. Maybe later we will have interactions, definitely, if you wish so. Okay, so yeah, with the permission of all our professors here, I'm just sharing my screen, and let me see. I'll share the whole screen, and then my presentation is visible.
Yes. Yes.
It's visible, right? Okay.
Yes.
Yeah, I have now kept it in the full screen mode.
Yes.
I hope you can all see that.
Yes, ma'am, we can see it.
Yeah, yeah. So yeah, so very good morning to all of you, and myself, Srija Lakshmi. I'm representing Indian Institute of Space Science and Technology. I hope our institute is familiar to a few of you. If you are not aware of my institute, this is just a 16-year-old institute located at the southern tip of our peninsular India. And we are under the Department of Space, Government of India. And along with that, we do have the autonomous status as well. So we are deemed to be university, supported by Department of Space. Okay, so being in the Space Institute, I was telling Pavitra and others that I teach a lot of engineering students, and teaching astrobiology to biology students, in fact, is easier. Okay, the reason being you have more domain knowledge than what I have. Okay, so we will start in a very simple, humble way. Okay, and proceed as and when we progress with the slides, in fact. Okay, so this is just a warm-up time. Okay, so I request all the students there, just try to attempt some things that I will show you, and you need not be very 100% accurate on what you say, but you should have something to say on each of the questions that I'm asking now. Okay, I hope all of our student community are ready. Okay, so yeah, I'm just moving one more slide. Yeah, this is actually a flash quiz. Okay, so you're all familiar with flash quizzes. Okay, this is astrobiology related flash quiz, very simple things, but we should be able to answer a few of them. So this is, I believe this question, natural process resulting in the evolution of organisms best adapted to the environment. Majority being biology students. Absolutely, I have no doubt that you are going to answer this. So you please keep all your answers ready, okay, for a self-evaluation, I would say. Okay, so the first question already is passed. Second, there is one abbreviation given here. It is LUCA. So can you please elaborate or expand this acronym? Okay, the hint is evolution process. Here comes the third question now. Approximately how many astrobiology missions have been conducted so far? It is just approximate. You can put it as under 20, more than 50, less than 100, or more than 1,000. Okay, so like some windows, you can put that. Okay, so it is not going to be anything accurate anyhow. Not everything is documented. Yeah, another one. We have one more acronym coming here, IPEX. So it was a very famous astrobiology mission. Can you please expand what IPEX is? Just the expansion for IPEX. It's a very famous astrobiology mission. Next question coming up. How old do you think the oldest fossils are? Okay, fossil study is one among the very, very prominent areas in astrobiology studies and research. So according to you, how old the oldest fossils are? Here comes the next question. You may be familiar with geological time scales. Can you please name them? I just want 4 of them, very major 4 geological time scales.
Ma'am, just to add to this, I'm their geoscience faculty. So I think I may have asked this during their exam as well. So I will also be interested to see.
Sure. The next question. What is the major distinction between prokaryotes and eukaryotes? As I mentioned, being biology students, absolutely, this is not at all a big deal. But very few engineering students, I mean, maybe 1 or 2 people are there, biotechnology, aircraft engineering students. So let them face this challenge. I think you already started the course, so you may be knowing all these things, but again, just refreshing. Yeah. So in astrobiology, another huge area is what you call as analogous sites, analog site research. So in India, as I understand, MIT is very much seriously pursuing some of the analog site research. So you can mention 1 or 2, just name them, 1 or 2 analogous sites in India, or maybe the state in which you can find analog sites. Yeah. So the questions, I'm just limiting it here. Okay. So we'll start just going through the answers one by one. The first one, of course, most of you must have done it right. The answer is natural selection. That is a process which is resulting in the evolution of organisms, and they adapt to the environment in that process. LUCA stands for anybody? Students?
Last universal common ancestor.
Yeah, last universal common ancestor, or sometimes it is LCA also. Okay, so I believe that you have studied the tree of life. Okay, yeah. And how many astrobiology missions approximately? The window between, is it less than 20? More
Maybe
than
10 to
100?
15.
15. 15, 1-5.
22.
22. Oh my God, very precise. Good.
More
Third
than
answer?
50.
Yeah.
More than 20.
More than 20. Answer is correct. But it exceeds certain other numbers as well.
More than 50.
Anybody of the opinion more than 100?
No.
Probably documented, no, but probably people might have conducted, we don't know. Okay, that is the beauty of astrobiology. We don't know anything. So sort of easy, but at the same time difficult. Okay. So in fact, more than 50 has been documented very thoroughly. Okay. I will show some of them, just the pictures, and okay, fine. And IPEX. What is the expansion for IPEX? IPEX was the intelligent payload experiment which was conducted by NASA to find whether CubeSat missions can be utilized to perform astrobiology experiments. Okay, so if you see intelligent autonomous experiments being planned on board CubeSat, that was the Apex NASA's mission. So how old do you think the oldest fossils are?
0.5 billion years.
0.5 billion years ago.
11.4.
11 point. My goodness.
3.5.
3.5
Life
billion.
originated. When did life originate? According to what we understand. It's okay, Kushi. It is okay. It is okay. No worries. Yeah. So approximately 4.2 billion years ago.
Yes, ma'am.
You agree with that, right? So typically oldest fossil around 2.5 billion years ago. That is what is documented. Okay. So you can take away 1 decade from your answer, Kushi. So that will give you the right answer. Okay. So geological time scales, you know them, right? So Hadean, Archean, Proterozoic, Phanerozoic. Okay, at least 4 I was mentioning. And among these, in which eon did the primary life originated? According to what we have the document now, which eon?
Primary life meaning the first life. Precambrian.
In this domain, I mean, Precambrian is within one of these eons, right? So when the bigger eon is given, anybody?
It's Phanerozoic.
No, it is Archean. Okay, first life originated. Okay, no worries. I mean, this is no way an evaluation to you. I told you, we are just warming up. And yeah, prokaryotes, eukaryotes, of course, biology background students, they know single cell, multi-cell, then without
Membrane
nucleus.
bound, non-membrane bound.
Yeah, without nucleus, with nucleus,
Yes.
cell membrane bound, not bound. So like that, no? Several distinctions are there. Okay, just this is the, at the outset, we call this distinction to be the most important one, or maybe visible one. Analogous sites in India?
Ladakh, ma'am. Ladakh.
Ladakh.
Gujarat.
Run
of Kutch.
Run of Kutch.
Yes.
And Rajasthan geological heritage parks.
Several are there actually. Okay, so I just put some of them. In Tamil Nadu, we are in IST, we have an analogous site research group who are exploring different parts of Tamil Nadu. Okay, so several states within our country have potential to be explored as analogous sites. Okay, so most of you must have scored above 5, right? Minimum. Okay. If
Yes,
not, it is fine. 3? About 3? All have about 3, right? Okay, because we are in the master's program, right? So we
yes,
need to have. So the thing is, your reading has to be a broader one. Okay, so you were trained in a specific domain, so you have to expand your domain in order to be a student of astrobiology. Okay, so I just wanted to confer that message. Okay, so before we start, okay, so we will proceed now. Okay, so this was just a warm-up, as I mentioned. So here, this is what we are trying to understand today. Okay, so I just put it as a formal slide, so that since it is recorded, later when you refer to these slides, you should know what context these things were done. So in this particular basic lecture to astrobiology, we are trying to find, along with others, we are trying to search for life in the universe. Okay, so when we talk about life in the universe, exclusively we are searching life beyond the blue planet. Okay, so throughout the lecture, the same set of questions asked in different ways will come up. Okay, so you refer to any astrobiology course or any astrobiology reference material, you would see one or the other form of questions being asked. And till now, 100% conclusive answers have not emerged for most of the questions. That further makes the branch of astrobiology as a very, very green pasture for research as well. Okay, so you have a very bright future in the research domain of astrobiology. That is one thing that I wanted to tell. Okay, and since you have taken up this very challenging area, of course, you are courageous also, that I would say. Okay, so we have certain responsibilities, right? So to learn certain topics, to disseminate the idea to a larger population. So all these things are now vested in your hands, okay, being trained formally as astrobiology students, okay. So the question, the biggest question, if you compile everything, every question of astrobiology into a single question, this is the one that I like the most. Okay, so here the question being asked is, are we alone in the universe? Okay, so whether the answer is yes or answer is no, both ways it is going to be scary. Okay, if you are alone in the universe, if the answer is yes, oh my goodness, what will happen if my year is or my time is gone? What will happen to the universe? And if you are not alone in the universe, again the same problem comes. Oh my goodness, somebody whom I don't know is there. That person, we don't know how they will look like, whether we can call he, she, or the constructs that we have created on earth, no, it may or may not fit into that particular species. Okay, so both ways it is going to be a tough thing. So along the course, whichever domain you are talking about, we are trying to find answer to this big question. Okay, so I will keep on popping up several questions throughout the course, and that is one of the uniqueness of this particular course, that also I have to tell you. Okay, so this is image from Apollo 8, which is the famous image Earthrise. I think you're all familiar with that. Okay, so before going into the details of the course, this is again something that you already have been introduced to. I'm just reiterating it regarding the history. Now we have a lot of resources to learn when we come to astrobiology domain. Okay, but if you trace back some 50-40 years back, the scenario was totally different. Even the word was not very popular. Okay, so while going throughout the course, you please try to understand how did we achieve the current state of knowledge that we have today. Okay, this is very important, because this idea of habitable world, the idea of some other organisms or some other living species in other planets, so what we call as Earth-like planets, all were predicted long back. For example, in the in 16th century, this Bruno. Okay, so philosophers were the prominent ones to start thinking that we are not alone, and then they were criticized, ridiculized. I mean, so many sort of things happen. But from that time of that philosophical, or rather we call a speculative science, currently this astrobiology has emerged as a sort of a science, an empirical science. And as you know, the empirical sciences are actually bounded by, or maybe constrained by, scientific experiments and the data generated. Okay, so now you cannot simply talk about astrobiology. You cannot simply put up any what to say, theories or anything. You have to contemplate it with data. Okay, you have to analyze it, and so many facilities are currently available. Space missions are dedicated to the study of astrobiology, and telescopes have emerged into high-tech equipments to gain data. Space missions dedicated, and with a lot of data coming back to Earth, we have a big problem there. The problem is with the interpretation. Okay, so how do you interpret the data from a space mission which is dedicated to astrobiology? It's a huge challenge. So I wanted you to be cautioned against some of the things that are pitfalls when we learn astrobiology. One among them is the interpretation of the data. Okay, because here, no, what we are desperately looking to find something, especially a trace of life, a trace of some remains that will tell that some life existed here or emerge here. So all these questions every time pop up in our mind that, no, if any data you get, you are very much eager to interpret it to some molecules of life. For example, water peak. If I get an IR spectrum, I'm always eager to locate that peak at around 3,450 centimeter inverse, so that I can locate a water molecule there. Okay, so at the same time, we have imaginations as well, right? So anytime I'm seeing something in a photographic image, okay, it may not be the accurate as a spectral signal. Okay, at that time, I'm very much optimistic about finding life. So there has a conflict. There is a conflict between that human imagination and that optimism and what to say, the lack of knowledge in interpreting certain things. Okay, and artifacts, whatever you see in such photographic things. So we have to be very much cautious that you should be having the ability to separate, okay, what our imagination wants us to believe. Because being an optimist, I always believe that, oh, I'm going to find the best in this particular experiment. But that is my wishful thinking, right? My optimism. And the data and the knowledge from the telescope and the missions, they may be talking something different, but due to my over enthusiasm, I try to fit my imagination into the data. Okay, so that can bring a huge problem. So this is one problem that you all students of astrobiology or are all practitioners of astrobiology should bear in mind. Okay, so this is a word of caution with which I'm starting the lecture. And on the right panel, what you are seeing is actually the Arecibo message. I think most of you know that there was a very eager missions to find whether intelligent life is existing beyond our blue planet. So people used to send waves, messages to outside to see that somebody will respond to us back. Okay, so although it seems funny, okay, people have made an effort. So this is the Arecibo radio telescope experiment. A lot of things were inscribed. So this is the what to say content which was originally sent. I think this image is very well known to you. You can explore this image further, and some of you, if you are interested, you can decode this particular image and what was the rationale that each of these fragment was sent to space. That is also an activity you can take it up and find out. So you will understand that the what to say, the desperate attempt to find extraterrestrial life or search for intelligent life outside. So a famous mission again by NASA, the SETI, S-E-T-I, may be known to you, that is search for extraterrestrial intelligence. Okay, so that was again a very famous mission. So several such missions have already been conducted. So as part of any of your minor project, you can pick them up and then find out why that particular experiment was done. Okay, that is one way of appreciating this particular language of astrobiology. And for school students, you have a very big responsibility to take these messages to the school students. Okay, so for them to make this course interesting, you should tell them such messages were sent, some gold foil experiments were there. So many experiments were conducted to in the effort to find extraterrestrial intelligence. Okay, so you just keep this particular thing in mind and be beware about the interpretations that we do. That's also important. Yeah. Then in any astrobiology course, it starts with telling that we have a motive or motto of finding life in other universe or life in other parts of the universe other than Earth. So the problem here is that, do we know what life is? Okay, and if we know that, why should that particular definition or why life should be a concern to astrobiologists? Okay, and if we need to understand life, of course, we need to find some characteristics that mark that life. So currently we have some classifications, living, non-living things, okay, and certain important characteristics of life that we attribute as on today is the life is very complex. It has properties like it can grow using some of the resources, it can metabolize, it can create energy, it can use energy, it can evolve into certain forms that can adapt to the environment, it can store informations, and it can actually synthesize molecules, or it can perform jobs, it can replicate. So all these attributes are in fact attached to the definitions of life. But here there is a caution. Okay, and for this particular topic, I would suggest that you can read one textbook is there. Okay, this textbook is by this is David Catlin. Okay, so if you can have that book, I have that book with me. So this is a very small, it's a primer. Okay, this is the book Astrobiology by David Catlin. If you can procure that book in your library, that also will be a good thing. Okay, it can be added to your reference list of references. Okay, David Catlin, the textbook Astrobiology, very short introduction. This can be a resource material for you, because it talks about several introductions regarding this search for life and all. So typically the book talks about whether these characteristics are unique to living organisms, and it tells that some of them are not unique because the complexity is not unique to living organisms. That formation of highly, orchestrated structures, even inorganic crystals can be grown. So growth, everything happens for non-living organisms as well. Okay, so it brings us to one big question, whether the definition of life is just a human idea. To some extent, of course, till we don't find another life, we have to assume that it is just a human idea. And even our microbial or the other so-called primitive life may have a better definition for life. Okay, but certainly to progress in a field, we need something like what you call as working definitions. So we proceed in this particular course with what we call as working definitions of life, because this is the maybe the best choice to proceed with a particular course like that. Okay, but at the same time, as astrobiology students or astrobiologists, we should also be ready to accept if we find life in other planets. It may be totally different from what we know or what we expect. Okay, so where do we astrobiologists look for life in other planets? We coined a word called habitable zones. Okay, again, the definition for habitability is with respect to the definitions of life, what we know. Okay, for us, it is the carbon-based life, it is the water-based life. Okay, the temperatures in certain domains, we need an atmosphere. So a lot of things are there in our definitions for habitable zones. So we have a lot of so-called Goldilocks phenomenon. Maybe in another module, you will learn more about the habitable zones and the Goldilocks zones. So here, of course, whatever you come across, please make sure that you are aware or you are able to demarcate between the science and sensationalism. Okay, so finding something in some other planet, it brings a lot of sensationalism, but it has to be supported with scientific data. So this is something that you should always bear in mind, because you may be proceeding with your research in the future. So this is one point that I wanted you to always lock it in your mind, okay, whenever you are dealing with astrobiology. Moving further ahead, yeah. So these are the sort of career options I would call, because suggestions about career path is something that you also may be having certain doubts. So this is something that I found from NASA's website. So they have that astrobiology strategy plan, where they are suggesting that since we several domain people come in the domain of astrobiology, you have that strong footing in your that level of graduate education, and you link the knowledge that you have gained in that particular domain to learn the certain aspects of astrobiology. Okay, and once you gain a degree in that, you graduate, then you start building the community. Essentially, this is the reason why I wanted to know how many students are there, because you 9 students here, you will form a community in the future. You will write proposals, bigger proposals, because every domain has to be covered in astrobiology proposals, right? And then you proceed together for funding support opportunities, and finally you emerge as a good research team, okay, who can survive on your own, and then you will end up in a career in astrobiology. Okay, so this is one way, because essentially moving from one's domain totally is sort of difficult, and we may not gain much out of such drastic moves. Okay, so please try to build bridges between your domain and the astrobiology domain, which has a close resemblance to that, so that you can contribute to the field in a very decent way. This is one thing again I wanted to tell, because this is just an introduction thing. Okay, so here I thought of talking to you a few topics, very simple topics only, aligning with the syllabus of your course, because your syllabus was already sent to me. So I thought of dealing with these many topics today. Maybe I will divide it into two parts if time permits, or else maybe we will have one more session later, because today I have some other commitments also later. So astrobiology has very important, what to say, footings on finding the definitions of life. So it is sort of bringing ideas together, then having conversations, and bringing up sort of a consensus. So this consensus is not because he or she is my friend. It should be based on some strong evidence. Okay, so in that way only we can build this astrobiology expertise within the country. And in the second point, we will talk about the origin and early evolution of life. Again, from several domains you can talk about that. But we will confine it into the simplest discussions for today. And the building blocks of life, of course, mostly being biology students, we are very much familiar with these things. I will just run through those segments. And in part two, there's a concept of habitability, but today I am not going to touch that particular habitability thing, but I will put a few theories of origin, just the outline of few theories. And the current searches of life also, this meteor part and all is actually not covered, but I wish that maybe in one more lecture I can cover that at a later part. Okay, so we will proceed slightly faster now. So what I mean, we are trying to find what is astrobiology and why should we have such a domain. Okay, so astrology is something very much popular. Okay, so we find luck around stars. So if you just introduce a word bio in there, we are searching for life around the stars. Okay, so here the one thing, where do we start studying this particular astrobiology? Of course, since we are searching for life in elsewhere in the universe, it is very much important that you trace or you study life which is already originated in some part of the universe. So exclusively, since we know that life has originated already on Earth, we will start our study from Earth. Okay, so the origins of life on Earth, the evolution of the life, how did it happen, and of course, can we predict something about the future of life? So these aspects of life are being studied in this particular broad domain of astrobiology. So everywhere, in several questions can be asked, and in every question you will find the key word as life. How does life originate? How does it evolve? What is the environment which support, or what can be the environments that can survive life? Are there any limits, or what you call as extremes, within which only life can survive? We have answers for most of them, most of these questions now, may not be conclusive ones. And if at all we find life in another world, should it be like what you see on Earth, or can it be slightly different? And is there, or has there been any other form of life elsewhere in the solar system? And even if there are any other forms of life or any other habitable world, can we identify such a habitable world, or can we observe them using the tools and equipments currently we have developed? And ultimately, the question is being asked is, what is humanity's future on Earth and beyond? Can we take this life to some other planets, and can we thrive well in another planet? Okay, so these are exclusively the space missions, current and the forthcoming missions are trying to understand. Okay, we are also part of some of the space missions of the country, upcoming space missions of the country. Okay, so the research of at the origin of life, about the origin of life, is the core of astrobiology. You remember, whenever you talk about astrobiology to anybody, you remember the question that the question that we are asking is the origin of life. Okay, so origins, evolution, and future. If we are able to understand these three big questions, yeah, we are done with astrobiology. Okay, but still, it's a very, very infant field, I can call it. We can actually call it. So these are some important questions. You should all remember that. Okay, so these questions will keep on popping up. Okay, and in IAST, in fact, I'm part of the basic course in astrobiology. It's a very simple 2-credit course for our undergraduate students. It's offered as a choice-based credit system elective. Okay, so we have 4 modules. The first one is the astronomy. The second one is the planetary geosciences. The third one is the astrochemistry, astrobio part, biology part. And the fourth one is the survival in extreme environments. So these 4 modules we cover in say around 30-35 lectures. Okay, and those who have interest in proceeding or finding what are the other courses which are available. So these are the 4 very interesting courses. I have completed all of them some time back. So these are offered available in the Coursera platform, okay, where the University of Arizona is giving the course Exploring Other Worlds, which is having an emphasis mostly on the astronomy part of that. Then from University of Copenhagen, they have the Origins, Formation of Universe, Solar System, Earth and Life. Again, that's a combination of astronomy plus some of the biology part. Emergence of Life, offered by Urbana-Champaign campus. This has a lot of things to do with the fossils and the biology part, and astrobiology and search of extraterrestrial life by Coquel, University of Edinburgh. It has a lot more of a chemistry component. So the point that I wanted to make is that you can frame up an astrobiology course with the domain expertise you are having. Okay, so you can put on your major and then you can elaborate the course on astrobiology. It is possible, and whoever is interested can proceed and take up the course and just enjoy that. They're very, very, very enjoyable. Okay, so now coming up the challenge. So since we are searching for life, the big question is, do we know what life is? Of course, there is a problem. So several definitions are available. According to Schrodinger, from a thermodynamic point of view, life is something that tend to approach the dangerous state of maximum entropy. So entropy refers to the dissemination of energy by systems. Okay, so when life is approaching a maximum state of entropy, it is called death. So if the life need to keep itself alive, it has to draw a negative entropy from its environment. So these things are something, I mean, beyond contemplation from a biology point of view. So we biology people or astrobiology people at large are looking at this puzzle of having a definition for life. And if we find anything, any body, how do you assign that whether it is alive in the first place? So we look for certain characteristics, as I was mentioning in the previous slide, including whether it's able to reproduce, whether it's able to enhance, I mean, grow, then can it utilize energy using the food it consumes and the metabolic processes, can it respond to environment, adaptation, ordered the hierarchical arrangement of cells or organelles or organs, these all types of things, whether they are available or not. So these are the things that we start looking. Even though we don't know whether these are correct or not, whatever we know from our earth-based life, we proceed with that to understand the features of life. Okay, so now in searching for astrobiological, from an astrobiological perspective, we are now comparing terrestrial life with that extraterrestrial thing. So of course there will be a small touch of cosmic what to say, belief or cosmic ideas coming in. Okay, so accordingly, several definitions for life. So this is actually part of your syllabus, definitions of life in the context of astrobiology. So some of them I have picked up and put it complete across, because when you see the slides later, you should be able to read and then start questioning for the completeness of certain definitions. Okay, so the NASA's definition, a self-sustaining genome-containing chemical system that has developed its characteristics through evolution of life on Earth. Okay, so this particular one thing is now commonly used one. So some other people call life as a genome's way of making more and more fitter genomes. So that adaptation, that evolution, survivability, all these things are being included in this particular definition. But we have a bigger definition, a broader definition, where life is, mentioned as a highly ordered system, which is carbon based. So I have emphasized this carbon based. So I have one more activity coming up for you in this particular domain. So currently we assume life to be carbon based, and it uses liquid water as the primary medium for chemical reactions. So making the life propagating a lot of millions of chemical reactions are coming into action. So these are all based, reactions are all based on liquid water, and the systems being built are carbon based. And this particular life is actually capable of processes. We name them biological, and some of them include metabolism, photosynthesis, replication, and of course the evolution, what we call as Darwinian evolution. And Earth being a rocky planet, we understand life which is being hosted by a rocky planet, and there is a nearly circular orbit which is happening around a normal star, and the star and the planet has to keep a distance in such a way that it should allow the presence of liquid water on its surface. So all these definitions for life, you can clearly understand that we have framed, we in the sense human beings have framed based on the idea and the knowledge that we have acquired so far. Now the question is, can we be very sure that the life need to be carbon based? That means, are there any other elements around which life can survive? Okay, one example is actually the silicon based life. So one of you can take up this particular aspect for your assessment or internal report or whatever. And can silicon be that particular element around which life can be, survive? And if so, can liquid water be there in this particular system? If not, what can be the other medium through which the life can survive? Okay. And yeah, coming, if people are interested in finding out the astronomy aspects of that, the planetary aspect of that, what is the significance of this circular orbit and the distance of the star and this particular planet in order to define the so-called Goldilocks zones? Okay, Goldilocks zones are the habitable zones. So these two things, either any of you can take it up and read and make a small summarized report, so that we can share it across the whole class and it will be very useful for you in the future. So this is these are another set of assignments or sort of activities. I don't call them assignments, they are activities you can pursue, and voluntary activity. Voluntarily you can pick them, choose them. Okay. So now if life is there, of course it started from something and we need to identify from where it started. So can we now go to the building blocks of life? Of course, we need to go, and some of them, again, based on the carbon-based life, water-based life, we are talking about here. And this is what you call as the periodic table. That biochemistry, chemistry students are very well aware of that. And in this biology, in this periodic table covering now more than 118 elements, we find 6 of them very much prominent in the life that we know. Okay, we call it as chnops, or in textbooks, several words are coined for that. Some people call it as a sponge. Okay, so here the priority was given to carbon, hydrogen, nitrogen. So I put it as chnops. So carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. These are the major elements that come up in the building of life. Okay, so now we know that there is something called genome. Okay, so if you combine all the genetic material in an organism, this is what you call as the genome. Okay, so currently most of the life is based on what you call as the DNA, the deoxyribonucleic acid. So this is what you call as the double helix of DNA, where you are having certain base pairs which are combined using hydrogen bonds. Okay, and we are having this this backbone made up of some sugars and phosphate. Depending upon the sugar part, you can have either deoxyribonucleic acid, or you call it another sugar is also nucleic acid is also there, what you call as the ribonucleic acid. So in the current advanced life, we find it is used built across using the DNA. Okay, so these are the purine and pyrimidine bases what you find in the DNA. Okay, so the RNA, we are having the bases. You see, cytosine, guanine, adenine, and uracil. Whereas in RNA, we are having this uracil component being replaced by another base called this thymine. Okay, so this fundamental distinction between DNA and RNA can also pop up as a question for your exam, because this is astrobiology course and we have very fundamentals to be covered in this particular course. Okay, so this is one thing that is very, very important. So remember, this carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur make 99% of all the living materials. Okay, so these are the fundamental building blocks of life according to our definitions so far. Okay, so before going further, there are certain key terms. So for biology students, it may not be a big deal at all, but usually I deal with this engineering students. So this biosphere, this is that particular layer of planet where the life exists. So this is the habitable zone in case of our system. Okay, so it comprises the total ecological system of the planet where the life can survive as well. Okay, and there is another term, barcode. So genetic code is an example, and genetic code actually helps us to distinguish between two organisms and also compare two organisms. Okay, so this is one thing. So the DNA, this part of the genetic code. So I already mentioned for earth-based organisms, this is the genetic biopolymer, which is very well established. So genome is the entire genetic code of an organism, and it has functions of replicating, then transcription and translation. So these three functions of genome is very important for the propagation of life. And if you are having many organisms, of course, we are interested to find the relationships between them. And this is done by the what you call as classification. And that particular classification, the study of relationships, is what you call as the phylogeny, where the genetic makeup of these organisms also are being considered. Next term is biosignature, very important, because whenever we detect certain species or certain materials, how do you find whether it is having life associated with that? For that, we go with what is called as biosignature, which is a detectable sign. It can be a chemical or morphological sign which supports the presence of life. RNA, of course, ribonucleic acid. Here, there's another thing, a prebiotic soup. Okay, so we believe that early life on Earth would have emerged with coming together of many elements which formed simpler molecules. Then they came up together to bring up complex molecules in the presence of water in the primitive Earth. So the mixture of all these components, inorganic as well as organics, are what you call as the prebiotic soup. And a progenote, that this is the single or maybe the initial person or initial life form which can act as the common ancestor for all life. So your Luca may be considered as the progenote in the case of the tree of life. So it was a free floating material, no cell wall, so it was not at all very well organized. But when time emerged, everything got organized. Okay, so continuing with the building blocks of life, you are very familiar with this particular ideas that all earth-based organisms use nucleic acids. So DNA, RNA, we were just talking about, and the hereditary information are stored or being transmitted through this DNA, RNA, and then proteins are being synthesized on the command, and these proteins also act as a catalyst. So they have a say on this particular biochemical reaction rates. And these phosphorus is also included in most of the biological systems, where they have the unique role of storing energy, and then ATP to ADP, that transfer is also happening, that phosphate transfer is happening, and energy is being released and then stored. Okay, so remember, from the simple or the single celled organism to the biggest mammal or the huge animals, complex animals, the same basic biochemistry is being utilized. Okay, so this is the one advantage when we talk about searching for life, because on Earth we find that everything started from a very simple single cell, and up to the complex organism there were some similarities. So there is something which is getting evolved in the process. So typically we will be able to, so this is the optimism that we are currently holding. With this experience in learning how life emerged or evolved on Earth, we may also be able to find some other form of life in other planets as well. So this is the optimism that we are holding. So when we talk about the origins of life, we talked about some of the raw materials that the earth-based life is currently having, right? So for bringing up this materials or this life, what can be the raw materials? So whether this raw materials existed on Earth or did it come from some other place, especially space? And how did the conditions became so conducive that these building blocks could assemble together to form very reasonable or some intelligent molecules, you can call them. Okay, and what principles did govern this assembly and this organization of matter, which didn't have any life, and it emerged into life, living systems, and that life itself started from a molecular level to a cellular level, then tissue level, organelle level, then you have the organism level, and further the ecosystem levels also. Okay, so did it happen? Did it co-evolve with the life as well as the biosphere and the earth? All these, did they co-evolve? This is another big question. And having said that, can life survive in very different environment? Or in another way we can put it, are there any limits of life? Okay, so these all questions will be answered or are being tried to get answered during the discussions in several astrobiology courses. Okay, some of the textbooks that I have mentioned over here, they are really interesting reads. As I mentioned, they also have some cosmic touch. Okay, so this is by a Belgian Nobel Prize winner, these first two books. Okay, they are available, I think, in Amazon. I don't know current availability. Just find them. If you are interested, you can make them part of your library as well in Amity, so that the forthcoming batches of astrobiology students can benefit from them. Okay, so it talks about a lot about the origins of life, and in the fifth miracle, actually, it talks something more about the chemical aspects as well. So these are some of the references or in fact, the resource materials which may be interesting for every student to read, so that you build a different your own perspective that can be further discussed with your friends and all. Okay, so so now the further question happening here is that where and how is a big question. How did life originate? That is one question. And before that, where did life originate? Okay, so several theories are being discussed, being criticized, being supported, so many areas. Again, this gives a the broad what to say unclear picture about the origins of life. So people support a view that there was something called a primordial soup, which was created as a combination of organic, inorganic components, and with some sort of liquid water. Probably the temperatures would be then supporting the liquid water existence. Okay, so it might have happened sometime after that particular sort of very high temperature Earth. Okay, and since high temperatures prevailed in Earth during those the prehistoric times, or maybe the time when life was originating, hydrothermal vents, that means that the fissures which are happening or available in the deep in the oceans, they are considered as one of the potential sites where the life must have originated. Okay, even there are discussions on the permafrost. Okay, the typical ice sheets which were available because the snowball earth was available at some point of time. So the ice sheets, can they be these starting points where these molecules all came together for the building of the complex life? Or some people even believe that life must have originated from outer space and got migrated. This is what you call as the panspermia concept. Okay, so several so-called hypotheses or assumptions are there with respect to the origin of life. Okay, so each one of them is a topic where you can take it up for a debate, because every single aspect will find debates, I mean, for and against. Okay, so scientifically debatable topics are available. But here, mostly the convergence have come that the life started, okay, or maybe the components for life started emerging in so-called the Hadean eon, because it is actually the 4.5 billion years ago, where the Earth was typically under hellish conditions. Okay, a lot of volcanic eruptions, dust, extreme temperatures, everything, so that no, uh, there's a whole lot of mixing up happened. So people tried to reproduce these conditions in the laboratories and then added a few elements so that to find whether the any biosignature for life can be synthesized in the lab. Okay, so of course there has been some success in such experiments. I will come to that in a short while. But then and there, it's a big question. So in the preliminary life, we were not expecting any cell membranes, right? But later on, the cell membrane started evolving. And what can be the conditions that actually prompted the formation of a cell membrane? So the lipid bilayers or many molecules, bigger molecules which can act intelligently, they also emerged. So what were those conditions? So being another one more important point is that being extreme conditions where we believe that the first life might have originated. So we have currently a big branch of study in astrobiology, what we call as the extremophile study. Okay, currently polyextremophiles are one good target areas to start your research or start your projects, because they give us a sort of important clues to talk about the boundaries of life. Okay, whether life has a boundary, and if it has, how does it survive till that outer boundary? Okay, so the adaptations of several life forms, everything are very important in the context of astrobiology. Okay, so these things are also important, we have to take into consideration. Then comes, yeah, so talking about the origin of life, I was talking about the Hadean condition, or the hellish earth, you can call it as 4 to 5 billion years ago. So the earth is still forming. The complete formation of earth has not happened after the Big Bang 13.5 billion years back. And the debris were there, the debris were hitting each other, they were generating more debris, volcanoes, everything made that particular earth as a hellish condition. Then we moved on. When we came to 4 to 2.5 billion years ago, it is marked that the era is called the Archean era, with several single cell organisms, and the first fossil microbes also got actually documented in this particular eon, what we call as the Archean eon. So the ancient life form actually is documented to be originated in the Archean eon. Then further coming towards the current time, eon 2.5 to 0.54 billion year ago is called what you call as the Proterozoic eon. So the oldest single-celled organisms and the great oxidation event happening, and I mean when just happened, and there was a rise of oxygen, so that no, there were organisms which get adapted to the oxygenated world, so that the respiration and the consumption of carbon dioxide by the planktons and the plants or the smaller organisms happened, and then the oxygen started being generated, and later on the organisms, this earlier life, got adapted to the oxygen environment. Earlier the oxygen environment was sort of toxic to them, and in the eon, this particular Proterozoic eon, the earlier life started emerging into the oxygen tolerant, you can call it as life. Okay. Then coming to the current eon, what you call as, including the Cambrian period, there are a lot of organisms, multicellular organisms emerged, and they actually radiated. Okay. So this is the term that we use in the context of radiation. This radiation, what I am referring to here is the thriving of the organisms. Okay, so it is not our the wave-based the physics radiation. This is something that the organisms were very well reproducing. They were happily living, and many diverse organisms also emerged as documented from the fossil records. So in a way, the Phanerozoic eon that actually saw the emergence of visible life in the planet Earth. Okay, so these four eons are typically important. Subdivisions are definitely available within each eon, and the extinction of certain species also documented. So those are other areas where you can actually frame up your internal assessment projects or something. What were the specific attributes of each of these eons, and what are important events these eons actually documented with respect to the origin of life? That is another very interesting topic. Okay, so I'm just touching upon the points where you can further elaborate. Okay, then coming to the several theories of origin of Earth. Okay, so among them, the famous one is what you call as the Oparin-Haldane theory. This is part of your syllabus, of course. So British scientists Haldane and Oparin independently, the biochemist is Oparin is actually a Russian biochemist. They independently came across this particular synthesis of organic molecules from the so-called inorganic or abiogenic materials. Okay, and the only condition was there should be a source of external energy, which was provided by ultraviolet radiations, because radiations were prominent in the early Earth. Okay, so it was since before this great oxidation event, Earth's atmosphere was reducing in the meaning that very less amount of free oxygen was there, and predominantly the Earth at that time contained gases like ammonia, then water vapor, then methane was there, hydrogen was there. So like that, some gases only were present. Okay, so both of these scientists independently they conducted experiments, because under the assumption that the first life forms appeared in the warm primitive ocean, where and they were heterotrophic in nature. So heterotrophic and autotrophic, two classification of organisms based on their food habits. Okay, so autotrophic, they actually get energy from the sunlight, and they photosynthesize and they do their own jobs, whereas the heterotrophic organisms actually they will use the nutrients which are available from their surroundings. Okay, so here this early Earth, they were having this particular heterotrophic organisms, and the compounds which are available on Earth became part of their diet. Okay, so the concept by Oparin is that the life developed from coercivate. So They are all some assemblages of some particles, and the microscopic particles then immediately they came together. They came together, formed some circular aggregates of so-called the biological molecules, especially the lipid molecules, and which were held together by electrostatic forces. Okay, so there were charged molecules, and these electrostatic forces held the molecules together. So they were having something, a sort of an assembly, and this sort of assemblages form the so-called the precursor for the cells. So here the Oparin further confirmed the formation of these coercivates, and these coercivates also acted as a so-called catalyst for biological or biochemical reactions, and in that way they were similar to what you call as enzymes. Okay, so these enzymes further they built the so-called membrane-bound cavities, and they can they can perform well in aqueous solutions using this membrane-bound materials. Okay, so and at this time Haldane was performing experiments. So he also he was not knowing about Oparin's coercivates, but he believed that simple organic molecules, simple molecules formed first, and then in the presence of UV, several reactions were triggered, which later led to the getting these molecules to become complex and ultimately forming cells. So there are several missing links between all the statements, of course, but something has to be taken forward, right? So these two ideas actually formed the later, it formed the basis of research on so-called abiogenesis. Typically that happened much later, after 1940s or 1950s. So the concept of abiogenesis is that the life arose from a non-life form more than 3.5 billion years ago on Earth. So this apparent Haldane theory is one important thing. You can expand it. Several resource materials are available which talks about the apparent Haldane theory. So I request you to elaborate on this particular theory further. Okay, it will be very useful. And when Oparin and Haldane was proposing these things, Miller and Urey, they went one step ahead by experimenting whether whatever they were conceptualizing can happen in the lab. Okay, so the first successful production of organic molecules from inorganic components, which were thought to have been present in the prebiotic earth. Okay, so those things were taken up. So we had the combination of they had the combination of water vapor, methane, ammonia, and molecular hydrogen were combined in the laboratory flasks. Okay, and it was pulsed with an atmosphere of electrical discharges, because in the atmosphere a lot of thunders, this thing, I mean lightnings, everything was there, no? So all the mixture was actually pulsed with electric discharge in order to find whether any molecules, simple molecules, can form. Okay, 1 week later, when the Miller and Urey was what to say investigating the flask, obviously they were in fact very happy to see that several simple organic molecules, including the amino acids, which are the building blocks of proteins, had formed in the flask. So typically it means that some sort of reductive environment with the support of an energy, big chunks of energy resource, might have supported the formation of in simple molecules. Okay, and these simple molecules further had some mechanism to come together and interact and become a bigger system, and that bigger system later became an intelligent system who can replicate on their own, who can make a copy and then transmit this information from one generation to another generation. Okay, so this became, Miller-Urey experiment became a very historically important experiment, and years later this Miller-Urey experiment was further simulated or maybe replicated in several other laboratories with very interesting results. So this is another area where you can just find what were the what to say continuing experiments with the Miller-Urey background, and even the shock or maybe this electric discharge, the intensity of the discharge even matters. That is what the latest publications are talking about. Last month also there was one publication. You can just search for Miller-Urey experiment and find how it was done. How it is being followed up by other scientists. Okay, so the consequences, of course, till the period when this Miller-Urey experiment was conducted, researchers have found, after this particular experiment, researchers have found that amino acids can spontaneously form peptides or maybe smaller proteins, and the nitrogen compound like the bases linked to sugar and phosphate groups in the nucleotides can form the starting materials for this prebiotic world. And this particular evidence that the RNA nucleotides can be synthesized at certain conditions, this supports what you call as the RNA world hypothesis. Again, this one is another major hypothesis in the history of astrobiology. And here, this particular RNA is actually, in terms of complexity, is lesser complex than the DNA, the current world. Okay, so since we believe that the initial life was could be a simpler one, it might have started from RNA. So RNA world hypothesis holds the idea that on Earth there was abundance of RNA life that were produced during using these prebiotic chemical reactions. Okay, so in a later phase, this RNA evolved into a strong sort of candidates for and acted as precursors for the DNA. So in that one important thing that I wanted to highlight is that RNA has a unique property that it can act as a catalyst also. So catalyzing a particular reaction, especially a biological reaction, is very, very important, because that particular guy can drive the reaction without being consumed. So whatever quantity of RNA was there, it will remain there and it will try to produce more and more RNA in multiple living forms. Okay, and that would have helped in the emergence of life, especially during the initial phases of life. Okay, so this RNA world hypothesis continues to be one of the leading self-replication first conceptions in the case of abiogenesis, and this is very well utilized for explaining the origins of life. It is actually one school of thought. Of course, like in any other branch, here also people have objections related to this RNA world hypothesis. We will just quickly look into the RNA world and the origins of life, and maybe in 5-10 minutes, and then I will stop for a while so that if there are clarifications, you can ask them, or else I will see whether we can proceed today or maybe later. Yeah. So before going to this RNA world hypothesis, this is something very interesting. Okay, so this is onezoom.org is one site where you will find a lot of tree of lives being created by people. You can submit your own tree of life. So the tree of life typically actually talks about the evolution of life. So how the life on Earth has emerged. It is typically the tracing of history of life on Earth. So from one generation to next, how the characteristics are inherited. So typically that talks about a simple life which originated and that contributed to the multiple forms of life that we see today. So once you are able to trace current life forms to this single root here, typically you will come to know what is the importance or how did the first life originated. Okay, so relatedness of forms can be done, and the amino acids and proteins are defined by the genes typically. So you identify them, genetic mapping can be done, and the difference in protein sequence will talk about the resemblance or the disparity between different species. Okay, so this is something which is the first, one of the first tree of life model, which is supporting the biodiversity. So this particular Haeckel's tree of life has almost 1,000 pages. Okay, so if you check into these websites and all, we will find the several tree of lives, and how one species to another, they are different, and how they are related, and how they can be traced back to a common ancestor. Okay, so Plantae, Protist, Animalia, this was one classification of tree of life earlier. Currently, it is a totally different tree of life. We will come to that. Okay, so coming to the RNA world hypothesis. Okay, so in order to find whether how the living organisms arose and evaluate in the evolution process, how do they express the hereditary information is the fundamental to understand that. Okay, so from the very simple life, today's life forms have become extraordinarily complex. In fact, we call them as machinery, and they can proceed from DNA to protein, and through an RNA intermediate, every synthesis is happening very effectively. Okay, so now the question is, how did this complex machinery evolve from such a small pool of some small molecules? So to support this formation, we are having what you call as the RNA world hypothesis, which actually states that the RNA world existed on Earth before modern cells arose. Okay, so typically the RNA world pre-existed even before the or maybe just after the first cell formation. The first cells with the DNA was actually evolving from the first cells with the DNA. Okay, so typically if we consider 15 billion years ago, where the Big Bang happened, and the solar system with our Earth was actually formulated somewhere in the between 5 to 4 billion years ago. There was the RNA world, and much before the first mammals were born, there was the RNA world predominating uh, the historic timeline for the universe. Okay, so again, molecular biology of the cell, this textbook is good indeed, and it talks a lot about the RNA world hypothesis. You can actually read. This is actually pretty slightly older book, but it talks in detail about that. And this RNA world hypothesis is the one of the popular. We call it as a pre-cellular life model. Okay, and proposed by Carl Woese, Crick, and Orgel. So they believe that the earlier life forms used exclusively RNA for the storage of genetic material, and the non-living chemicals could not have given rise to cells in a single step. Several intermediate life forms must have coexisted, and under certain situations or certain circumstances, they came together and joined to form their current complex cell. Okay, and the Crick actually proposed the RNA as the primary genetic material, since it has the capability of self-replication. Okay, and exclusively that self-replication property came due to its ability to act as an enzyme. Enzyme is a catalyst, so it can promote reactions, and it can assist in the self-replication. So and one more important thing is that the same genetic material RNA can also be converted to the complex DNA by the process of what you call as the transcription, reverse transcription. So those people with biology background, you may please tell or discuss in your class what do you mean by this transcription, translation, replication, all these stuff. From a biology point of view, you are much familiar with these things to discuss and then bring out whether this is possible or not. Okay, so the hypothesis that RNAs could have been the initial materials which were contained in the cells are very strongly held with this RNA world hypothesis. Going further, RNA has very important uniqueness that it can store both genetic information and catalyzing the chemical reaction. So this is something that DNA can do, but this is something DNA cannot do. Okay, so these two roles were equally done by RNA, so this became an important molecule. Okay, the pre-cellular or cellular early life. Later in evolutionary time, it is emphasized or it is highlighted that DNA took over the genetic material, and then proteins were made, which became the catalyst and structural components of the cell. So RNA world hypothesis again is a very, very important topic that you have to learn in detail, okay, according to the syllabus that is there for you, okay. And here, this is one important highlight about RNA, what you call as the autocatalysis. Okay, because life has to progress, right? So every time it has to multiply, it has to adapt, it has to do so many functions. So the autocatalysis is something that might have enabled the life to reach the current status. So the first biological molecules must have formed on surfaces of minerals because they were not having any life. And then the molecular synthesis and the metabolism, even the chemical reactions could have existed long before the first cells arose. And when cells all came together in sort of a confinement, these all processes might have got inducted, and in some way life must have started. That some way is still to be addressed, okay. So one important thing that is to be emphasized is that definitely life to propagate required molecules that has the unique ability to catalyze reactions. It can be either direct interaction or indirect interaction, but the ultimate result of that interaction is to have more and more molecules being formed, similar to the one that is already existing. So with more and more molecules with this catalytic property, definitely the mass of molecules, the mass or the quantum of molecules must have increased, and that might have led to the formation of life, okay. So catalysts, of course, have this particular property, and if we use them as raw materials, of course, they will act with very sufficient specificity, as well as they will create more molecules without much of damages, or we call as defects, okay. So what type of molecules? So several such molecules in the prehistoric time might have that autocatalytic property. Okay, so another area of research where people are looking forward is that what all category of molecules might have that property. So polypeptides are one candidate, strong candidate, because they are still available. They are still doing this job in the current time. But the problem with polypeptide is that although they are good, versatile as catalysts, they don't have that ability to replicate. So there is a problem suggesting polypeptides as molecules with autocatalytic property. So what can be the next candidate? The next candidate is polynucleotides. Of course, they have this information storage and catalytic properties, okay. So with these two properties, you can have the exact copies of their sequence. That is why DNA, RNA splitting is happening. Complementary base pairing enables that there is no defects happening and the copies are getting equally formed. And one polynucleotide acts as a template for the formation of other, because this splicing, this splitting happens and joining, splicing happens. So this templating mechanisms are complementary in action, and this is lying at the heart of DNA replication and the transcription which is happening in modern day cells, okay. But here, if nucleotides have to be synthesized effectively by templating, extra catalysts are required. So in the absence of catalyst, of course, this templating can happen, but it will be very slow. More importantly, it can be error prone and inefficient. But when we consider the life's evolution, we find that the polymer formation would have been faster, it has not much of errors, and it would have been efficient, which suggests that this polynucleotide may not be the ideal candidates for this autocatalysis. So the big question is now, before DNA and RNA polymerases, which can catalyze these reactions, who could have catalyzed these particular formations of these biological molecules and cellular materials? So now, then came the greatest, one of the biggest discoveries, which actually fetched the Nobel Prize in chemistry. which happened with the discovery of so-called ribozymes. So earlier, people had the belief that only proteins can catalyze chemical reactions, cellular reactions. But Altman and Thomas Ketch, they discovered so-called ribozymes. Okay, so ribozymes are typically a class of ribonucleic acids that are also capable of catalyzing chemical reactions. So now we have somebody other than this RNA or within the RNA world who can catalyze chemical reaction as well as initiate this particular branching or maybe the catalyzing and producing of more molecules. So the strongest argument in support of this particular ribozyme as supporting the RNA world hypothesis is that the ribosome, which assembles the protein inside a cell, is itself a ribozyme. Okay, so this ribozyme is actually the landmark discovery which supported the RNA world hypothesis. So here the statement is a full statement is made. It is going like this: despite the fact that ribosome is composed of both RNA and protein, the process involved in translation are not catalyzed by the protein, but by the RNA, indicating that early life forms must have used RNA to catalyze the chemical reactions before they started using proteins. So current world, DNA world is being supported by proteins as catalysts, but in the RNA world, there were no proteins, but we were having this ribozymes, which acted as the catalyst. So this discovery actually supported or supports the RNA world to a very, very vast extent. Okay. So now one more school of thought, whether pre-RNA world predated the RNA world. Of course, there are some indications for that also. And it is very difficult to imagine the formation of long RNA chains purely by enzymatic methods, because non-enzymatic methods. Non-enzymatic means there are a lot of errors which can come up. Okay, but precursors of RNA, which are the ribonucleotides, are also difficult to be formed non-enzymatically. So many statements are given here. So here we try to tell that before the RNA world, there can be a pre-RNA world. Okay, and this is the RNA which is having now this base, the sugar with the pentose sugar. Here it is actually a hexose sugar when it comes to the pre-RNA world. And there's one more molecule coming up that we will discuss later, what we call as polymers, which are similar to or RNA-like polymers, which are the first biopolymers with both information storage and catalytic activity. So polymers that resembled RNA, but chemically simpler might have been the precursor. So you see from here to here to here, it happened for the evolution of RNA material. So for building this RNA material, we believe that simpler precursors must have been very crucial. So these all things are contained in some of the reference textbooks that I have mentioned. So please go through it and have notes on your own. Okay. So this lab experiments were also conducted where the simple forms, the polymeric nucleic acid can act as the template for the synthesis of RNA molecule. So the first RNA molecule was formed, then it diversified gradually, took over the functions of the synthesis, the replication, as well as catalyzing the reactions, and then it emerged to the current RNA world. So this is the way in which the things emerged as far as it is documented. So from single strand, the double strands were built, and it happens through several mechanisms. Okay, so sometimes bulging happens so that this particular area will be exposed to some other moieties. So three centers, single center, it all attract the complementary chains together. Sometimes loops were formed, the new strands can be formed, similar internal loops. These are all secondary structures telling that there were several secondary and tertiary interactions that led to the formation of several new RNA molecules. Okay, so elaborate structures, highly complex structures could have formed with the support of several of such interactions. Okay, so these are all having significance with respect to so-called non-covalent interactions or electrostatic interactions in some cases. So it is anticipated that such types of secondary, tertiary, even quaternary structures and interactions might have resulted in the formation of highly complex structures. So these are all important things. So here comes one activity for you, which is an important activity because RNA world hypothesis can be either proved or disproved. Okay, so you use chemical evidences. Lot of reactions are involved. I didn't mention the reactions in detail, but using chemical evidences, you can actually prove or disprove that RNA arose before the DNA evolution. Okay, so this is something that I would like you people to take up and complete. Okay, so that will be important, and this is a very important reference which actually talks about the RNA world. So this is a puzzle piece we are having here, a full picture of RNA world. So it starts from prebiotic chemical synthesis, ribozymes functional, then evolutionary theory, then you are having this cooperation between molecules, complementarity comes here, then we are having energy metabolism, earlier environment, and replication of RNA. So this is a very important reference. It should not be missed. Okay, so this is Nature Reviews in Genetics, which you can read, and this will talk a lot before you meet this particular puzzle joining activity. Okay, so you refer to all these things and let me know if you need any further support to understand it, or maybe having discussion or assessment. Okay, so before before closing this segment, this is something, evolution of life is also an important aspect in understanding this astrobiology. So you very well know this person, Charles Darwin. So Tree of Web project. So this is a very important project. You can just refer to this site and see out all ways it helped in bringing up discussion platforms. So we find similarities between several of this particular branches, and this commonly they emerge into bigger tree of life. Okay, so the broken branches will be the crust of earth, and this branches will keep on continuing with life evolving into more and more advanced life. Okay, so if anybody has studied this evolution in tree of life, you know that initially the tree of life from the Luca, we had the single cell prokaryotes and the multicellular eukaryotes, where this membrane was not there in the case of prokaryotes, whereas the eukaryotes were organisms with confined cell membranes. We have currently come to a tree of life where the branches are not two. In fact, this is a subsequent development. Now we have a three branch. So the archaea, eukarya, and the bacteria. So we have this three branches here in the modern tree of life. So this paradigm shift in the tree of life happened due to this particular scientist, Carl Woese. Okay, so a British scientist. So here, from two branches to three branches. Earlier it was archaea and eukarya and bacteria. So this archaea got added to the domain. Okay, so how did this similarity is mapped or marked in this tree of life is by gene sequencing. Okay, so I want you to refer to this word molecular clock, okay, which is used to mark the resemblances between the various, because all of them meet at a common point, right? So there is something that is unique which makes them part of a single family. So this is done using sequencing, whereas 16S ribosomal RNA is the gene used as the molecular clock, because this particular gene is conserved to a large extent in a lot of species. Okay, so most of the domains that you find in the tree of life will have this particular gene. Hence, it is being used as the molecular clock. So Carl Woos was that particular person who made that landmark change in the tree of life, which is typically a paradigm shift in a tree of life. So in the oldest books and all, you will find only 2 branches or different branches. Now we have this particular branch as well, and you can locate several prokaryotes as well as eukaryotes along with the archaea family. Okay, so these are the distinct features of pro and eukaryotes. It is very well known to you, so I'm not explaining it further. So as I mentioned, if you look into this particular tree of life, you see that they are related evolutionary, they are related. So their biology, some sort of biology has been retained and some has been different. So they are classified into different branches. Okay, so the common ancestry is being indicated. So here comes the common ancestor here. If you are able to find who was here and how and when he or she was created, the person, the organism or the life form was created, you are very much near to the identification of the origin of life in the universe. So with that indication, we may start searching for life elsewhere in the planet. So locating this LUCA or LCA, last universal common ancestor or the last common ancestor, is a very important step in the understanding life from an astrobiology perspective. Okay, so yeah, I just stop here because I'm not proceeding with the theories of life now. Okay, so hopefully maybe today I have something more. If you have any queries, definitely we can have some discussions, and I hope because it was a monologue, I need to complete it before 11:30. That is why I didn't entertain any questions in between. But of course, I hope that you got something, I mean, minimal something from what I was talking about. We will continue with the theories of origin of life, maybe at a later, some 7-8 theories are there. Okay, so I'm really sorry that I cannot find today time because some other things are already lined up. Okay, so yeah, any queries? I can refer to some of the reading materials because I have given a lot of reading materials in some of the slides. Okay, so all the students, I'm requesting you please go through the materials. Okay, because your astrobiology study is largely research oriented. Okay, it's not a single textbook oriented. You cannot call it like this because, as I mentioned last week, there was a report on the Miller-Urey experiment. Okay, so that the pulses of electric shock that they are giving, it has impact on the synthesis of these amino acids. 22 amino acids were created at another laboratory. So every point I was trying to connect as much as possible because it's a very vast area. In 1 hour, it is very difficult. If I take the RNA world hypothesis, it will go to 1.5 hours lecture. But I tried to sort of capsulate it everything into a smaller thing. Yeah. So students, did you benefit to even a certain extent? I'm eager to know that so that no, we can.
Yes. So, first of all, thank you, ma'am, for taking 2 hours of your time to give this very insightful talk.
Yeah.
And I know it's very hard to comprise and cover everything within 2 hours. So we can always, as you suggested, and if the students are also equally interested, maybe think of a part 2 if that is something that they are interested. I'll ask them to get in touch with me. Any of the students, if you have any questions, you can put it in the chat box, or you can also unmute yourself. As you know, Dr. Shrija Lakshmi likes to interact a little bit face to face as well, so don't be shy. Okay, so ma'am, Renish has put up a question. If we are trying to look for biosignatures, we will be looking for compounds similar to Earth-based life. Then how can we detect life which might have evolved differently?
Exactly. So I didn't talk about biosignatures. The biosignatures are all chemistry, Renish, as you appreciate it well. But the problem is, again, the biggest problem with astrobiology is that we are looking for something whose definition or whose makeup we don't know, that we have to admit. Okay, so that is one criticism as well as one challenge with astrobiology. Because if I'm looking for life, as you have rightly mentioned, we will be looking into biomolecules that we know, like RNAs or DNAs, or maybe the premature forms of these things, amino acids, this and that. But if another form of life is existing in somewhere else, or maybe even coexisting with us, we are unable to identify that life. Okay, so that is actually one drawback of astrobiology, this field, and drawback of the biosignatures. The extent of molecules that we have classified, it can be isotopes, it can be small molecules. We classify biosignatures into some groups. I had that slides with me today. I couldn't share with you. Okay, so this is a problem. So rightly saying, we cannot identify such a life with current knowledge that we are having. That is the answer. It is not possible. This answer as far as current knowledge is concerned. The second part of the question, if we say that life could be evolved differently from here, why can't we see life thriving in nearby planets like Mars? Yeah, of course. In Mars, from even the Viking landers, if you remember 1976 onwards, people were having that clue that, okay, Mars may be inhabited or not. But even today, with a lot of other rovers missions, a conclusive evidence for life in Mars is not there. Okay, people are very near to telling that, but we are not that courageous enough to announce that we found life, because we are not sure of that. Okay, so Mars may be a very prominent or a promising place to find a life. Okay, that is one thing, yeah, to answer your question, because conclusive answers are not available as on today. Okay, yeah.
So ma'am, just to follow up on that question, do you think, for example, sulfur based or any other compound based can be?
Again, it is being hypothesized that, if you excuse me for a
Yes,
one
sure.
Let me find where I can, yeah. So yeah, I'm sorry about that. Sulfur is another potential form of life, element which can support life, and lot of research articles are available with respect to that area also. Because as we mentioned, even the silicon based life, phosphorus based life, sulfur, but they all have some limitations at one point or the other. Because the having this much of diverse chemistry without the support of an aqueous medium. Okay, so that is somewhere everybody is sort of, they are falling off there. Because this diversity in chemistry, which can be brought up by this carbon, especially due to its catenation property and not, sulfur has similar properties. But with respect to the stability of the molecules that sulfur can form, vis-a-vis the stability of the molecules that carbon can form, we have certain sort of a disparity there. Okay, but obviously in some other places where conditions will be totally different from our Earth conditions, it may be even thriving now. We are not able to identify that. That is what Renish was asking. How do you know that? Yeah.
Okay. Yes, Mayuri, do you have a question?
Yes, ma'am. Sorry, I was unable to say. Hi, ma'am. I was unable to say hi to you before, so I wanted to say hi.
Yeah, you were trying. Yeah.
Yes. My name is Mayuri. I've done my bachelor's from Ria College in Life Science. So my question follows with Renish. We don't know that what other life forms or what other type of life might be there. But if we take our life as an example, so what could be the molecules that we look for, or any signatures or anything particular that we look for on other planets, assuming that it's the life similar to ours. So what could be those specific molecules?
Yeah.
That
Typically, Mayuri, if you have gone through the recent research articles, there are several organic molecules that are detected in the interstellar medium, ISM. Okay, so in the interstellar medium, what you call as the interstellar clouds and all, several organic molecules are available. They can be as small as, say, a nitrile molecule, a cyano-containing group, hydrogen cyanides, or maybe sort of many aromatic compounds are there. Or if you can find any amino acids, if you are aware, there are researchers happening in our own laboratories, where the amino acids are being created in shock tunnels. So we take molecules, certain elements, and then give shock to them, for that amino acids will be created. So similarly, what is being done is that you take elements that is known to you, you bombard them with several forms of energy, or you provide them with very extreme conditions that you anticipate in other planetary bodies, in order to see whether something that we can detect is being formed. Okay, so several molecules can be taken, amino acids can be taken as biosignatures. So everything one thing you understand is that whatever the composition of this Darwinian, we call it as the Darwinian pond. Okay, Darwinian pond is another term that you can search for. In the Darwinian pond, only the primary things happened. And from there, what you call as the prebiotic soup. Okay, so any molecule in the prebiotic soup is a prominent or a potential biosignature for that matter. So in that way, trace out the research articles, several molecules you can find out. But again, the limitation is with respect to our understanding of life, our definitions for life. Okay, so yeah, that is important.
Right, ma'am. Thank you. Just one more small question I have is that with the formation of a particular planet, affect the process of these molecules being formed?
Of course, because the chemical bonds, when they are forming, they are directional in nature. Because if one carbon and one chlorine is coming, and one carbon and oxygen is coming, the carbon will choose the person who is having a higher affinity. Okay, so that bond will be formed in the direction where that chlorine or that other atom is sitting. Okay, so the orientation, or maybe the topologies in which two molecules are approaching, that were also very critical. So that is why we don't know how come these molecules understood that I have to combine with the other person. That is totally a dark area, gray area, you can call it as. But somehow the atoms or the systems were intelligent enough to identify the right partner. Okay, so those things we cannot explain without any cosmic thing. That is why this cosmic ingredient is also put in astrobiology. So after a while we tell that somehow it came, but from a science point of view, that somehow is not allowed. But for astrobiology, we deliberately put it. Okay, so there is no other option for that. So orientation and the approach and the distance through at which they approach, because it has to be less than angstrom units, right? Then only the bond formation can happen. So several things are there, and recognition of certain species when it comes to the self-assembly or all those things, complementarity, cooperativity, all these things actually matters.
Thank you, ma'am. Thank you so much. It was a wonderful lecture.
So we have a question from Pallavi. Could life have originated in deep sea hydrothermal vents where conditions are similar to those on early Earth?
Yes, yes. In fact, in the theories of origin of life on Earth, the highest vote is for deep sea hydrothermal vents. Okay, and this fact is further supported by the tree of life, where you locate a lot of extremophiles in the certain branches which originated earlier. Okay, so definitely the hydrothermal vents, deep sea hydrothermal vents, are one among the potential regions or maybe locations where life might have originated. Again, there are certain what to say, lot of people support that, but there are certain objections for that theory as well. Okay, you can find it out, and if you don't find the resource, definitely you can just mail me back. Yeah.
Yes, we have a question from
Virus. Yeah. So virus, in fact, no, since we already know that viruses are nowhere in the tree of life. Okay. But one thing we know is that, according to us, a virus becomes a living species only when it is attached to somebody living, right? So according to my understanding, if viruses are to be detected, we need a biology already running there. Otherwise, the virus does not have any existence, right? So typically, as other chemical molecules, maybe it is possible to detect them, but I'm not sure about how to, I mean, in what way they will be interpreted. Any data, if how can we rule out whether it is from a virus or not? That I have to refer to. I'm not sure about that.
Yes, Aditi, you have a question?
So my question was, when we think about Schrodinger's definition of life, it's basically like the entropy of the universe is a stream, and life is trying to get or trying to swim upstream. So what was the necessity for this chemical moieties to undergo replication, or what was the necessity for them to self-replicate?
Yeah, that is according to Schrodinger's definition. Life is trying to increase entropy, right? Okay, because if the single life is there, if it is trying to increase its own entropy without replicating at all, it will face that it will vanish. Okay, but the life was trying to dissipate the energy. Dissipation of energy is typically what is referred to as entropy here. Okay, so it is not the entropy, the term that we used to hear from our school days onwards is disorderliness. But here, by the usage of the word entropy, in this context of definition for life in the astrobiology domain, we mentioned that the life want to dissipate its energy to the maximum number of similar life forms. So in that context, definitely replication was one of the ways, right? In other ways, it cannot. I mean, it will perform. Maybe another form of life must be doing whatever you are telling also. Okay, but our life form, it got sort of advantage. It got its advantage by dissipating this particular energy in the form of replication. Yeah.
Thank you, ma'am.
So, Dr. Pavitra, can we stop here now?
Yes, definitely. Thank you so much, ma'am, again for giving 2 hours of your time. If the students have any other questions or concerns, they can get in touch with me, or I'll share your email ID with them as well.
Sure, sure,
if they
sure.
have any concerns, and I will get in touch with the students to see if anything else is required from their end. But thank you once again.
Thank you, thank you. The pleasure was mine. So students, all of you, please make sure that you read maximum number of articles, and your study is going to be a research-based study. Okay, and some of the concepts that I was just taking few more time, you have to make sure that you study them, because exam point of view also certain topics are important. Okay, so in case of any support from my end, please let me know. Okay.
Yes, definitely.
So I hope you benefited to at least to some extent. Yeah. Okay.
All right. Thank you, ma'am.
Thank you. Thank you so much. All of you.
Yeah. Bye bye. Maksud ji?
Yes, ma'am. Can I end this?
Yes, yes, you can end this.
Okay.
Thank you.